All right, let's get going. This is 5.5 on cancer immunology. So let's just start with the basics. What is cancer? Cancer is the result of multiple DNA mutations that cause normal control of cell growth and death to be lost. So you get unregulated growth and replication. Just some good introductory stats for you. 20% of deaths in the industrialized world are caused by cancer. Median age of 70, so this is typically a disease of the elderly, though not exclusive to the elderly. Treatment, we usually see a combo of surgery, radiation, and cytotoxic drugs, aka chemo, commonly referred to the slash, burn, and poison method of treatment. A really cool note here is that most of us have had cancer cells develop in our bodies without becoming aware, because the threat is typically dispatched by our immune system. So think back to CD8 T cell function. Think back to NK cell function. Both of those are very interested in MHC1 and the presentation of intracellular antigens. And if something is virally infected or if it's tumorogenic, that is going to change how things are presented. So your immune system usually gets rid of cancerous material. So thank your immune system. We have approximately 10 million strand breaks a day in our body. So a breach of the DNA. Think about our place in the greater cosmos. The sun is beaming down solar rays at every waking hour of the day. The cosmos itself, we're getting bombarded by cosmic rays. Yes, the atmosphere protects us a good bit, but we still have these ionized particles breaking our DNA at regular intervals. But this is usually not a big deal because we have reparation mechanisms. There's a bunch of DNA repairing enzymes within the nucleus. And furthermore, most mutations are harmless. Most of the time, if you replace an A with a G or with a T, you don't see many. You don't see a change. You don't see a really damning issue. So a lot of things have to go wrong. You have to get a mutation in the right spot, usually multiple mutations in the right spot, and the repairing mechanisms have to falter. What are some genes we're worried about here? First up, proto-oncogenes. These are genes that contribute to normal cell proliferation, so telling a cell to replicate at the right time. When these are mutated, we get an oncogene. So the normal functioning version of this gene is a proto, so like a precursor to an oncogene, which is weird that we have this thing named in our functional normal physiology cell, something named after cancer. But I think this really reiterates why I bring up pathologies in this class, because a lot of the routes we use to discover some of these concepts and these genes and these cytokines are from the diseases they produced. So when we see this gene get mutated, we usually see, or genes in this family rather, get mutated, we see cancer. So that's how some of these things get their name. Tumor suppression genes, same kind of deal. When these get knocked out, we see tumors. So that's how they got their name. These are proliferation suppressors. So when I don't need proliferation, when I need that to stop occurring. When mutated, we have an increased cancer likelihood, right? If cancer is just unregulated proliferation and unregulated growth, if I have a gene that's controlling that gets knocked out, well, we're going to get more cancer. This is crucial to know. P53 is a hallmark tumor suppressor gene that is mutated in over 50% of cancers. It is a real smoking gun. Okay, so not concerned with the number, but just know if this is mutated, your cancer likelihood just went up by a substantial amount. All right, let's get going on this awesome slide here. So these are all beautiful picture representations of the core characteristics of cancer. Okay, first, they stimulate their own growth. So I have this sunflower watering itself. They secrete cytokines that aid in cell proliferation. Makes sense. The tumor wants to be alive. It doesn't know what it's doing is pathogenic in nature. Ignoring growth inhibition. It doesn't know that being big and pressing on the physiology of the surrounding area is a bad thing. So it runs the red light. It says, I'm good. I have these self-stimulatory signals to keep me alive. I'm going to ignore any inhibition. In the same sense, it avoids apoptosis. So if I'm getting these survival signals, I'm ignoring inhibition, I'm not going to die. I'm going to dodge all the bullets sent my way. And then lastly, and something I brought up in class because I just learned some more importance around this, angiogenesis. So think about the irregular anatomy. Things are proportioned exactly how they're supposed to be. If a tumor in a tight area gets really, really large, well, you only had X amount of blood vessels to supply that area. And now you have, let's say, 10x the amount of tissue because of tumor growth. So what happens is the tumor actually secretes compounds, one of them called VEGF. You don't have to know it, but just to have it here. That will induce angiogenesis, which is the formation of new blood vessels. So angio means blood, genesis means to create. This is a real hallmark in cancer and tumor generation, because the angiogenesis that actually occurs here is subpar. It's shoddy. These vessels that the tumors create are leaky in nature, so we tend to see some edema, some leaky fluid around the tumor, and that's really important for identifying things with MRI, with CT, because you see a ring of fluid around some of these tumors. So a really cool concept to understand. Angiogenesis will follow tumor creation, and the angiogenesis done is poor. Metastasis. So that's represented by the forest fire here. A big thing with cancer is it likes to spread, and that's what metastasis is. After we cause that big chunk in the local environment, we usually send off another satellite to go build up a cell population elsewhere, which is not good. Constant replication, so we have this copier here, and immune evasion, very good at hiding from the immune system. So the immune system is constantly doing cancer immunosurveillance. I think that's a really cool concept that I can't stress enough. So that's why when we saw with some of these viruses that we talked about in the middle of the semester, when they mess with immune function, your cancer risk increases heavily because the immune system was combating cancer this whole time. You just didn't know it. And another note, it is hard to truly eliminate these cancer populations due to all of these factors, as well as the fact that cancer cells like to form little stem cell populations. So if you don't get 100% of the cells, which is in all likelihood impossible at the microscopic level, you run the risk of remission, of these stem cell populations being capable of producing another tumorigenic body. Okay. Then we have tumor-specific antigens and tumor-associated antigens. So they sound very similar, but let me break down the difference because it's important. Tumor-specific antigens are structures just not found on normally behaving cells. So not related to cancer, but viral proteins. If something is infected my cell, and now I have like a COVID spike protein popping off my cell, well, my immune system is going to say, hey, what the heck is that? Mutated cell proteins, that's more so cancer. So when the cancer has caused the machinery of my cell to be so mutated that I'm now presenting markers on my membrane that are also mutated. Another example of this is amino acid splices. So if protein binding and chemistry in the cell is going completely haywire because of the cancerous mass, we can just end up combining random amino acids and creating these just nonsense markers on the cell membrane, which are not supposed to be there. So that's why I have this swamp monster mutant here. This is a smoking gun. If these are present on the cell membrane, obviously, if there's a mutant on the cell membrane, something's wrong, right? This is a dead giveaway. Something a little more sneaky that requires a little more immunology knowledge are tumor-associated antigens. These are found on normal cells to a lesser niche degree. The best example I have for you here are testes antigens. So in healthy individuals, these are expressed in the immune-privileged environment that is the testicles, okay? testes antigens, if they are expressed anywhere else, that would be a problem. Those are supposed to be sequestered, hidden away, only in a specific environment. But in the cases of cancer where DNA is getting read haywire when I'm expressing proteins that should not be expressed whatsoever, then we run the risk of potentially presenting a testes antigen where I absolutely should not be presenting one. I don't know, say in your neck or your brain. I don't know. And the correct or it does beg the question: can this happen in women? And absolutely. You have those chromosomes exist those genes exist on the X chromosome. So or maybe not on the X, pardon me. They are within the genetic capacity of the female as well. So yeah, that would be pretty alarming to your immune system, especially for women to express testes antigens. So very funny. I have the Among Us imposter here because that's like, all right, something must be amiss. Although testes antigens aren't inherently pathology. They aren't inherently bad. But when I see them on like my internal jugular vein, I'm just using that as an example, that's not good. All right. Another mechanism here, immune evasion. Okay, cancerous cells are really good at downregulating MHC1. So that's really important because that's how the CD8 T cell understands that something's wrong. So very smart by them to get rid of that tag. 1/3 to 1/2 of cancers have defective MHC1 expression. So that explains why some of them persist so much. And now you might be thinking, if you are astute in remembering all of the cells we've talked about, if MHC is gone, that's missing the self marker. And I have a cell that's supposed to deal with that. My NK cell. These detect missing self. So what are they doing in this? So a transformed or a strained cell, in this case a cancerous cell, totally strained, abnormal, that will express MIC. MIC, which is just like, it's literally just MHC1 that's tweaked. I forget the acronym again. I forgot it in class, I forget it now. So imposter MHC1 is the best way I like to think of it. So NK cells are going to bind this MIC because that is the incorrect MHC expressor. So now I know, okay, if that's being expressed, then something's wrong. NK cell binds it, gets rid of that cell. Cancer cells figured this out too. I don't know the means of how this is figured out, but cancers can cleave this MIC marker that lets my NK cells know when something's amiss. So now my NK cells are rendered inert because they lost their tag that was telling me when something was wrong. All right. Another crazy layer to this. So I'm just compounding all the reasons why cancer cells and why tumor bodies can exist for as long as they do. It really does weaponize the immune system and evade the immune system in really creative ways. So remember, our Treg cells are arbiters of peripheral tolerance. They have the tumor environment gives us heavy preference towards Tregs because that brings an immunosuppressive environment around the tumor. So now we have dysregulated resolution. Our Tregs are really good at saying, all right, the infection's gone, the inflammation should go away now. We need to rest and relax. But in this case, that tolerance is mighty improper. Right? I have a whole cancer body forming in whatever region that's being affected. I need my immune system to get rid of that now more than ever. But my Tregs are confused by whatever compounds the tumor is secreting, selecting for more Tregs in the area. So we talked about autoimmunity a good bit this unit, and that was a failure of Tregs to be suppressive. But now this is a failure of Tregs being suppressive, but at the wrong time and place. So an important distinction. And this is really physiologically important too, or clinically important rather. The more Tregs in the area, the worse the prognosis. And on the opposite end of that, CD8 T cells, the more of those present, the better. Because remember, CD8, that's getting rid of those intracellular issues. So ideally, if that is the major cell population nearby, then I have a higher likelihood of getting rid of whatever is causing me trouble. If I have a lot of Tregs around, they're going to cause anything that's trying to help, they're going to tell that to calm down. So that's the idea there. A really cool therapy that is in development, I think it's being used, but it's constantly in development, are CAR T cells. So these are chimeric antigen inhibitors. What we do is we take the patient T cells out and mash the TCR with the light chain of an IgG antibody. Okay, so I have this antibody here. We take the light chain, which has pretty good high antigen binding specificity, and we yoink it right here. And then this bottom half is the T cell receptor's intracellular portion, so the part that's steeping into the cytoplasm. Antibodies are better at tagging antigens. T cell receptors are better at transmitting the signal into the cell. So we take the strengths of both and we make our own super soldier. So now I have a CAR T cell that has really potent binding affinity as well as as a excellent intracellular messaging. So the TCR-BCR combo is amazing. That's why I have the griffin down here, combo of a lion and an eagle. Get the best of both worlds. Now, is there such thing as a too robust T cell? Something to think about. Absolutely. Absolutely. So that's something we can run into risk. We have the likelihood or a potentiality of creating such a powerful T cell that it might react to the self. So there's a lot of care and give and take with these, but it is a really cool concept that I wanted to at least introduce to you guys. So not a cure-all by any means, because we can make too robust T cells, but it is really, really cool that we can take our soldiers out of serum and beef them up to hopefully aid in the patient's survival. And then monoclonal antibodies making their way back. So we talked about these with type 3 hypersensitivities, and these are really common treatment pathways these days. I think, I don't know what the numbers are, but at least anecdotally, I see these drugs ending in AB all over the screen. And now hopefully after this class, I've ruined you and you will see them as well. But they're used, we administer these antibodies to target, in this case, cancers ailing the patient. So if there's a specific type of tumor that's expressing a specific type of receptor, I'll grow up a bunch of antibodies in a lab that target that receptor. And then hopefully I can get rid of that mass. What's cool about these is we can also load the FC portion with whatever the opposite of a care package is, right? With a lethal package, a toxin, a drug, or a radionucleotide. So really, really cool here. Something to just pour maybe enzymes onto the tumor mass, maybe a chemotherapy drug, or we radiate it directly, maybe decreasing some of the cons that come with radiation. I did look it up after class. Commonly tagged with iodine and yttrium, though I'm sure that's not an exhaustive list. But yeah, so imagine we have a big old iodine, an isotope of an iodine atom hanging off here that's a bit more radioactive in nature, that can hopefully disrupt some of the replication that the tumor cell is exhibiting. So yeah, really, really cool. I think this is a awesome development in pharmacology, weaponizing the immune system as a mechanism of action to help protect the immune system or ballast the immune system. So really brilliant stuff. All right, and then finally, we have immunodeficiency and cancer. Increased rates in patients with HIV, right? HIV, very immunosuppressive, kills your CD4 T cells. That's bad news bears. Organ transplant, I likely will be on immunosuppressants for a good chunk after getting a new kidney. And then obviously, if you're born with no thymus, or if you're born with some sort of congenital issue, yes, that will increase cancer risk. And I mentioned in class, this is how we discovered HIV. We saw a really high increase in Kaposi's sarcoma, I believe in some neighborhood in New York, and it happened to be traveling in a homosexual community where we saw a high incidence of this typically rare cancer, and that was due to this community having a higher incidence of HIV infection, which was harming their immune system, causing cancer protection to be decreased. Now, again, unfortunately, that led to a lot of homophobia around the case, but, treatments are getting better, and hopefully that homophobia is also waning. But yeah, so a real excellent example of the interplay between different biological fields. And we have an increased risk following infection, again, with oncogenic viruses. So there's that HIV example. HPV is a big one, highly related to cervical cancers. So that's why those vaccines are pushed early on, even potentially before being sexually active, because on the chance or whenever that does happen, why not have protection against something that could lead to cancer is the idea there. EBV, we keep seeing that virus pop up. So after having mononucleosis, we have a lot of risks because of that altered, that virus changing your B cells, your memory B cell gene pool. I also have Hep B in here, but I don't have the lay down for you on that one. But it is just important to understand that some of these viruses really do increase your risk of developing cancer due to messing with proper immune function. So it's something that I never really considered, especially early on in my biology career, that the immune system is so heavily related with preventing and removing cancer at the earliest stages. There's a high likelihood that a lot of us have had it, but the immune system has dealt with it. Toot sweet. So again, drink your water, get moving, get your lymph moving, get some sunlight, and thank your immune system for probably saving your life umpteen amount of times by now. Alrighty, that's all I have for you folks today. We have a talk on vaccinology next, and that is the last video of this class. Crazy stuff. Thank you for your continued attention. Adios.